Okay, so welcome to the second breakout session of the day and the last breakout session of the day. And I'm really excited about this actually because we've done something that we learned about from other rare disease conferences where they do poster presentations, not just from researchers, but from families. And so we're going to kind of go back and forth today and have researchers do some presentations and families do some presentations. And I think that's really great because we know that the researchers are also, families are also experts in a lot of these things as well. So looking forward to some great conversations. And so our first presenter is Sylvia Okafor. Okafor, sorry, from Dr. Amy Ramsey's lab at University of Toronto.
And I use this to move forward? Yes. Okay. Good afternoon, everyone. I'm Sylvia. And I'm a PhD student in the Ramsey lab, where I've been characterizing the hematology in mouse models of GRIN1 disorder. Typically, when you think of the NMDA receptor, you associate it with the central nervous system, but they're also important within the periphery as well. And despite being found in lower levels than in the brain. Okay. Recent studies have found that the NMDA receptor is important for iron homeostasis and blood cell development and function, as well as blood cell homeostasis, all via the modulation of basal intracellular calcium. We intend to identify blood biomarkers that can indicate disease status by investigating blood cell properties and whether they contribute to symptoms of the disease. So far, we've identified several biomarkers of disease, as well as for delineating gain of function versus loss of function for the GRIN1 knockdown mice. So this is which is a model for haploinsufficiency. Okay, there we go. We've identified. Oh, sorry. Okay. So for the GRIN1 knockdown mice, which is a model for haploinsufficiency, we've identified changes in iron content as well as within white blood cells. And we've also saw atypical blood cell volume and blood viscosity. Proposed mechanisms contributing to these abnormalities include reduced Gardos channel signaling and reduced iron homeostasis. And in conclusion, I believe these mice have reduced red blood cell deformability and and they have iron depletion. For the Q536R loss of function patient variant mouse line, we've seen changes in blood cell volume, iron content, blood viscosity, as well as blood cell proliferation. Again, proposed mechanisms contributing to these abnormalities include reduced carteles channel signaling and reduced iron content. In conclusion, I believe these might have reduced red blood cell deformability and early signs of iron deficiency. For the Y647S gain of function variant, we saw signs of polycythemia as well as increased blood viscosity. And for these mice, they likely have increased Gardos channel signaling as well as increased JAK2 STAT3 signaling. And in conclusion, they have increased red blood cell deformability and potentially polycythemia vera. In conclusion, the hematology, histopathology, and rheology of these variants suggests that the GRIN1 loss of function variants displays iron dyshomeostasis and reduced red blood cell deformability. And the GRIN1 gain of function variant displays polycythemia or potentially polycythemia vera and increased red blood cell deformability. I hope I've convinced you all that hematology is important for GRIN disorder, and that's why it's impatient to that's why it's important to investigate patient hematology. So the next step is to to collect blood samples from patients and compare the results to our mouse models in order to see if the results are phenocopied. And so please come on my poster if you would like further discussion and details. Thank you.
So we have to keep things moving today, but we want to give everyone a chance to at least have one question. So is there anything in the room?
Thank you. That's very interesting. Do we know what subtype of NMDA receptors are expressed in the blood cells? Obviously, you've got the GLUN1 subunit.
So, there's all subunits are expressed in. It depends on the blood cell type. So within red blood cells, all of them are expressed except for, I think it was GRIN2D. I'm not sure which one, but I know it's one of the GRIN2s. And for platelets, 4 of the subtypes are expressed. I don't remember which ones, but I know GRIN1 is. And for white blood cells, all of them are, yeah.
Thank you.
Okay.
Okay, our next speaker is Gilliane Vanderleeuw. So come on up, Gilliane. I just want to say, like, that was the great conversation or a great presentation. And, you know, one of the just little things when we met in 2019 in Atlanta, I think most researchers really thought that only GRI genes really expressed in the brain had any impact, had any functional role. So even just that one part of the presentation is pretty profound, that it confirms that they play a role elsewhere.
Well, hello, everybody. I'm the parent of a 15-year-old daughter with GRIN2B. And since 2020, we have started her on L-serine, and we've made some very good progress with that I would like to tell you something about. Oh, no, that's not what we needed. Does this work?
Oh, that's great. What are you going to do?
Oh, no, I'm going further instead of going back. Could you please restart it for me? Sorry.
You know, at me, not the.
Oh, that's the idea. Sorry. Okay, there goes my 5 minutes. Okay, well, L-serine is an amino acid, which is one of the building blocks of protein. It occurs naturally in foods like corn, soy, turkey, chicken, eggs, but your body actually produces it itself as well. And if you don't produce L-serine, you actually have another metabolic disease called L-serine deficiency. The active form of L-serine is D-serine. And at the laboratory level, it was discovered that D-serine makes neurons with a loss of function mutation work better. But D-serine is unfortunately not safe to give to patients. So they give patients an L-serine form, which in the body is then changed into the active form D-serine. Okay. The first clinical case for L-serine was actually conducted by the Barcelona GRIN team. That's David Soto, Dr. Mireia Oliveira, and Dr. Javier Alto-Valladillo. They studied the L-serine in the case of a little girl with a GRIN2B LOVE mutation as well. And meanwhile, a lot of studies have been carried out worldwide. And basically, it proves that L-serine is a very safe drug, has little to no side effects. There can be some digestive issues like diarrhea or constipation, but that's about as bad as it gets. And by reducing the doses, that can be most of the time very easily remedied. And meanwhile, children are successfully treated with L-serine in the Netherlands, in Germany, in Spain, and in other European countries, and also in the US. Go, please. Dr. Garcia Cazola, which is a metabolic pediatrician in the Centro de Geo Hospital in Spain, Barcelona, has conducted the first clinical trial for L-serine, and she has included 24 children, 6 GRIN1s, 5 GRIN2As, and 13 GRIN2Bs, which means that L-serine is safe and applicable for all GRIN mutations with a loss of function. And the results of this trial are most likely published this summer. And what are these results? At our last GRIN Europe conference, she gave us a first peek. Attention span, reaction time improve, executive functioning improves, children learn to imitate and engage in imitative play. I'm not going to read everything because that won't work time-wise. Children understand double meaning, the ability to interact non-verbally improves, sleep improves, and fine motor skills improve, which leads to better drawing skills and writing skills. A lot of these things are subtle and are hard to capture with the standard tests that we have right now, like the Bailey and the Violent. And that's why currently in Barcelona, they're also developing a GRIN scale, which is specifically dedicated to GRIN, so that these more subtle changes can be caught as well. Okay, hearing all of this and learning about L-serine, my husband and I lobbied very hard for our L-serine trial in the Netherlands, which we finally got in the Radboud Únase in Nijmegen and AMC in Amsterdam. It was a limited number of children, approximately 5, and it was under supervision of Dr. Clara van Karnebeek, who's also a metabolic pediatrician. And that trial is also finalized meanwhile. And that's the one our daughter participated. That's my girl. And we started her, as I said, in July 2020, 4 doses of 8.2 grams a day, so 32.8 total. And that's based on 0.5 gram per kilo body weight. And the first 2 weeks, you give half of that. It's very easily, you dissolve it in tap water or in still water. It's tasteless, so you can drink it without problem. And we personally buy our L-serine from the Serine store, which is an American company. It's FDA approved of pharmaceutical quality, and very important, it's affordable because L-serine can cost up to 250 euros per 100 grams. What did Charlotte see? She takes more initiative. She's better at learning and remembering routines. She is better at planning and can devise a plan to carry about simple tasks. So her executive skills have definitely improved. You can literally see the little wheels in her brain turning. She makes connections better. She can also relate to things she's learned a long time ago. Her focus is much better. She's better regulated. Go please. She's more alert, takes more initiative, no more zombie modus, which is a very important one for us. Sleep has improved a lot. Reaction speed, fine and gross motor skills have improved. And she jokes a lot more, understands sarcasm, double meaning. And of course, that's all very subjective. That's a parent's hope. But fortunately, we're very proud to say that we also have objective effects of the L-serine. During the last 2 intelligence tests, her IQ has improved. The gap between her and her typical peers has not increased, because first she was set off against 13-year-olds. Now she was set off against 15-year-olds. So you expect her to fall further behind, but she hasn't. She stayed at the same level, and she even managed to reduce the gap between her developmental age and her calendar age a little bit. Also, her MRI and the neurological test showed improvement, and her PRPP interview showed significant improvement. So we're very happy to say that L-serine definitely worked for us. Thank you for your attention.
Any questions?
Thank you very much. Very nice. Is your daughter taking any other medications right now? Sorry, I missed the beginning of your talk when I came in. I apologize.
Well, yes and no. She takes paracetam, which is a drug basically developed for Alzheimer and for dementia, which increases focus, concentration, and memory. But that's about it.
Thank you very much.
You're
And I'll
welcome.
say that we're just going to do one question per presentation, but if there's time at the end and presenters are still here, we can add more later. So our next presentation is Jinyeol Lee from the Colin Lab at the University of Toronto. And I'm going to turn this so it's a bit easier for you guys to see the slides while you talk and still be on the mic.
Is it a PDF or is it a presentation file? It's a PDF. Okay, yeah. So like how do I skip? What's laser? Oh, so it's working. Okay. All right. Thanks everyone for coming. My name is Jinyeol Lee. I'm a PhD candidate in Dr. Graham Collins Lab, affiliated at University of Toronto. So the main focus that we're doing and now for the research is to characterize the phenotype of the GRIN1 G629 variant mouse in particular, that we probably would know it's the identical mutation, the variants that Bryson, that our president Keith's son is having. So the aims and the approach that our research is focusing on is basically as a first part that we try to characterize the impact of the GRIN1 G629 variant on the NMDA receptor function and synapses and the behavior. So we generate this mice that is mimicking the variants that have been identified from the children. And not a lot of study has been done by using a mouse model. So we created this mouse in 2020 that I remember correctly. And then we wanted to look into the physiological, physiological complexity of how this is affecting in their, not only in the NMDA receptor function, but in synaptic physiology and how they would behave if they're like following the same principle of the children are exhibiting. And that's kind of part one of the research that we're focusing on now. And a little bit of spoiler that we almost reached the end of this point. So we're preparing for a manuscript that we're going to publish it soon. So it's going to be open access by hopefully in the next couple of months. And the second part, as we mostly went to the Dr. Amy Ramji's talk in this morning, that the part two is going to be establishing the potential therapeutic strategies using the genetic rescue experiments to restore the NMDA receptor functions that is getting back to where it normally is supposed to be at. So sharing about the research part as the part one that I just mentioned. So we're trying to hitting as many as different angle possible to get a better understanding of how this variant is impacting this mice. So we generated this mice, as I just said. So we're looking into synaptic physiology using electric physiology. So I wanted to see how the biophysical property is exhibiting when the G629 variant subunit GLUN1 is incorporated the functional NMDA receptor. And we're looking into the molecular biology level. So we wanted to look into the protein expression level and also how it affects in the other genetic networking, mRNA or DNA as well. And to have the advantage of having this mouse model, we're also looking into the behavior assays. So we're looking into the innate behaviors, whether they are moving it properly, they're reacting properly in their environmental cues, are they showing a decent motor functions? And one of the key experiments that we're doing it now, I think it was in the survey from the Gilliane Foundation, that the cognitive function is the primary symptoms that the relatives and the family are expecting to rescue. So we look into the cognitive functions, and I'm going to show it in the last slide of today's presentation. And the anatomical analysis, so we want to look into their histology, into how the brain is developing in this mice. But today, due to the time limit, I'm going to show about one slide of the electrophysiology and the cognitive part on the behavior assay. So just a quick showing that the phenotype of the GRIN1 G629 mice is pretty much following the same principle of what the children have showed. So on the description, there were described it to be hypotonia and light-weighted. And it's showing the same both in male and female. This has been taken a photo at age of 10 weeks old mice. They are definitely shorter. As you can see, they are much smaller in comparing to the size, both in male and female. And the weight is also being light in the variant mice. And the cognitive functions that we measure by looking into the spatial recognition memory. So we close this arm specifically on one side and allow this mouse to travel in between of the starting arm and the familiar arm. And after about an hour late, after about 15 minutes, they get exposed to the environment. And an hour later, again, we did a test trial opening this blocked part so we can see whether they could recognize this is a new space they wanted to explore into. And we saw that this mice doesn't really try to enter in this arm. So it kind of reflects that they are having a little trouble recognizing the spatial terms. And not only with that, we wanted to look into their recognition to the novel object. And after 15 minutes and an hour after exploring to these two Lego that we used, and we replaced it one to another different type, color, tech Lego, and it was showing that they couldn't really recognize the novel object. So they are having a deficit in the cognitive functions. Try to make it quick. And then we also looked into the NMDA receptor function because it's in the mutation of the NMDA receptor. They didn't really show much of NMDA receptor mediated transmission, as you can see it here. And quite surprising that they don't really have like a trafficking deficit. So even though this variant subunit gets expressed, they still get into a tetrameric receptor and reach to the synapses. So the conclusion that we have it in now from at the moment is they get expressed pretty well. They get incorporated like oligomerization into a tetrameric receptors, reach to the synapses without having a deficit in the trafficking. And then, but at the synapse, they just don't work as the normal wild type would do. So that's the conclusion that we have it reached at the moment. Some more details are at the posters, so please feel free to visit. And I'll be happy to take any questions now and even at the posters. Thank you.
Anyone have a question? Okay, we get lots of questions outside.
Later. Yeah.
Yeah.
Okay. Thank you very much.
Thank you. Okay, next up is Denise Rehner.
So this is not going to be a scientific presentation. I might argue that it might not even qualify for basic, but a very school for visual impairment in preschool 2 days a week, and then public school 2 days a week. And it was not very long until I realized it was a very stark contrast between the learning environments and what they were able to provide in the public school, even though they assured me that they were qualified for my son. My instinct told me they really did not have the experience, and they did finally admit to me they had never had a child as impacted as my son, which was a little bit heartbreaking for me because as a mom, he's my only child and he's all I know. And so for me, it's easy because he's all I know. But I created these booklets if you wanted to pass them, for example. So this was not my own effort. It was a team of his private outside providers that I invited into the school to observe and help write recommendations. Okay, so there's a lot of slides. I'm gonna go through it really quickly just to give you an idea of the content that I included. And the feedback that I've gotten from his team and from new caregivers, that is, the information is very helpful. And parents have asked me for copies of this. I've uploaded it to the GRICon, and so you can download it, and I'm happy to share it with you. But it basically goes through basic medical history, safety information, likes and dislikes, some of his challenges and strengths, concepts and words that he's working on. The optimal learning environment. A lot of our kiddos have cortical visual impairment. So in the school system, they don't necessarily know how to teach with kids with CVI. So his communication style, his mobility challenges, things that he's working on in his OT play skills, all of his therapies, his eating preferences, and then obviously emergency contacts. So just basic GRIN1 medical information, safety, things that are really concerning for us parents, like knowing that our kids always put things in their mouths. So just warning those really common things to watch for. if your kid has any seizure activity, any activities around, any safety around outdoor activities. I actually had this written into his IEP that any new team member for the school has to get a copy of this. Otherwise, you know how things fall through the crack. And anyway, so likes and dislikes. A lot of our kiddos like this common things: vibration, lights, music, etc. Things that he likes to do. He likes people. He's very social. Some of his favorite toys. Some of the strengths and challenges, you know, these reports that we get from the schools. I just included some of those things so everybody's on the same page. Some of his strengths, and these are pictures that we've gotten from different therapies that he's attended, different camps for special needs, just showing examples of how to engage him. Some of the concepts and words that he's working on. So a lot of our kiddos are nonverbal. A lot of them are working with AAC technology. So it's important for them to use common language, the same consistent language that we're using for repetition. This is a lot about the CVI learning environment. So our kiddos need real tactile objects that are vision friendly. So I took a lot of the recommendations from his outside providers, and so I use that to tell them, it's not me telling you what to do. This is a professional. Again, the CVI, important things to include. Speaking his language, just meaning using really relevant words to him, narrating his world. Like for us, it's easy to see. Well, I'm out of time. Like, did you hear that buzzer go off? That means I'm out of time. That type of thing. So strategies to keep him engaged, his communication, nonverbal, you know, watching for some of his nonverbal cues, some of the goals that he's working on in his therapies, his mobility, PT goals, examples, to give them ideas what they could do. These are OT skills, eating, or some of our kids are, you know, there's tricks and that moms only know or the parents only know, and then obviously emergency contact information. And that's it.
Question for Denise?
I can send you the PowerPoint.
Can you make them for others?
So I guess this is like IEP. Like, do you update the goals every year?
Not so much, because the goals don't actually change that much.
Okay.
But I do take all the goals from all the different providers and kind of merge them into one.
Okay. This is so helpful.
Oh, good. Thank you.
Okay, I'm pretty sure, I don't have it up in front of me, but I think Megan Sullivan from is up next. So Megan is also in the Ramsey Lab at University of Toronto. So shout out to University of Toronto. Good showing here today.
So hi, everyone. I'm Megan Sullivan. I'm a PhD candidate in Dr. Amy Ramsey's lab. And today I'm going to be presenting a bit of my work about the characterization of the GRIN1 Y647S heterozygous patient variant mouse model. Oh, okay, so just a bit of background information about this variant first. So the individual carrying this variant presents with seizure, limited verbal communication, and intellectual disability. You can see this variant occurs in the transmembrane domain, kind of depicted schematically there. And in vitro studies have pointed to this variant causing an increase in NMDA receptor activity in response to glutamate and glycine, so the two activators of the receptor, making it preliminarily a gain of function variant. But of course, more studies are kind of needed to validate that. So the aim of my work has been to validate this patient variant mouse model as representative of patient symptomology, as well as potentially a predictive model of patient drug response. Oh, let's go. Okay, so first I'll just provide just kind of a brief snapshot of some of the alterations we've seen in our mouse model so far. So just looking here, it does appear that both male and female Y647S mice have a significantly reduced body weight compared to wild type counterparts. And then here we have looked at kind of the convulsive behavior in these mice. And the Y647S mice display kind of the tonic-clonic events, kind of the muscle contraction and falling to the side, as well as kind of the absence-like events where there is a loss of awareness. And we did a 12-week handling paradigm with our adult mice. And you can see kind of the frequency of the Y647S engaging in these convulsions seems to increase with age and handling, but those are kind of two very difficult to separate things when you're working with mice. The next test we looked at was a way of looking at muscle function. So essentially we placed the mouse kind of upside down on a wire cage top and then just measured the latency to fall to the ground. So how long they can hold on. And the Y647S mice had a significantly reduced latency to fall. So they do kind of have that muscle loss of muscle tone. Next, we looked at a social interaction test. And essentially in this test, the mouse is kind of allowed the choice between a social stimulus, which is an age and sex matched kind of wild type mouse in a cup, and then an identical cup that's empty. So we can use this as kind of a looking at the preference of the mouse for either a social interaction or not. And what you'd expect in a wild type shown in gray here is that they spend significantly more time with the social stimulus rather than the non-social. And we did see that with the Y647S mice, where they are spending significantly more time with the social stimulus rather than non-social. And surprisingly, we actually saw that they are spending significantly less time with the non-social stimulus. And you can see they're kind of trending towards spending more time with the social stimulus as well. So it seems that the mice do have a hypersocial phenotype. Lastly, we looked at ultrasonic vocalizations, or USVs, and this is a form of vocal communication that mice elicit in certain contexts. So the context that we looked at was a male mouse being presented with a female mouse. So the male will emit kind of these ultrasonic vocalizations in a mating interaction. And you can see that the Y647S mice did produce significantly less calls in response to the female mouse's presence, indicating that they may have kind of communication deficits. Lastly, we looked at in vivo electrophysiological response to patient medications. So essentially, we can do this by implanting electrodes in brain regions that are relevant to seizure, sorry. So in this case, the frontal cortex and the hippocampus. And according to caregiver report, the sodium channel blocker lamotrigine best controlled the patient's seizures, while cannabidiol exacerbated them. So these are the two drugs that we wanted to look at in the mouse initially to see if the mouse kind of responded to the medications the same way that the patient had. So first, looking at the results of the frontal cortex, frontal cortex here. So we have ictal rate on the y-axis versus the drug that we tried, and then the post-administration time point. Kind of the most interesting finding from this graph is that lamotrigine at that 5 milligram per kilogram dose did seem to be potentially trending towards a reduction in the vehicle, which is our control. And then most interestingly, in the hippocampus, once again, the ictal rate and then the drug that we tried, you could see that 100 milligram per kilogram CBD, so the highest dose, which would kind of be in line with what a patient might be receiving to control their seizures. It actually significantly worsened the ictal rate in the mice. So it's kind of reminiscent of what the caregiver saw in the patient. So just in conclusion, our mouse is kind of displaying behaviors that are similar to the patient's symptomology, as well as potentially presenting a drug response that mimics the patient. So this just kind of validates our model as valid and just allows us the opportunity to kind of dive further into the pathological mechanisms that may be underlying these differences, as well as give us confidence that when we do kind of find a medication that may work, that works in the mouse, that it may work for the patient as well. Thank you so much.
Thank you. Question.
Sorry, very interesting about the social part. I'm wondering if what you're seeing is lack of fear to a strange mouse too, more than the social part, just because we've heard a lot of families describe that the kids have no fears.
Yeah, we've actually done a test looking at kind of anxiety-like behavior where a mouse has an opportunity to be in like open arms or closed arms. In a typical wild type mouse spends more time in closed arms just because of that fear of open space. In these mice, we actually saw they spend more time in the open arm. So it does kind of point to kind of a lack of fear or like a daring type of behavior. Yeah. Thank you so much.
Thank you. Okay, so our next presentation is Calvin Horn, and you may recognize his voice from every time you go to the GRICon website.
Hi, thank you. I want to say that this presentation is not really going to be scientific. I just want to use this time to kind of give you a snapshot, talk about our daughter, Kendall Hom. She has GRIN2D. Just to give you a snapshot of what she's gone through the past 7 years of her life. So at the birth, she was a preemie, born several weeks early due to a difficult pregnancy. She had irregular heartbeat, so she came out early. And at 12 months, we noticed pretty much a global delay, developmental delay, at which point our doctors said, hey, let's do a whole genome exome genetic testing, and we found that she had GRIN2D. There you go. So in January 2019, my wife, Kylie, started GRIN2D Facebook group trying to connect with other families. And through early intervention and therapies, Kendall started showing pretty good progress. She started to eat orally. She could sit up on her own. This is at the age of between 2 and 4. She took steps on her walker, and then also through connecting with other families. We were able to join the first Cure GRIN conference. I believe that was in September 2019. Correct me if I'm wrong. So that kind of gave us a lot of hope. At the age of 5 is a turning point for her. She suffered a grand mal seizure lasting over 24 hours. I think it was about 25 hours. And so she was put in a medically induced coma. And after 3 unsuccessful extubations and several meetings with the palliative care team, we decided the only way to get her back home was to have get a trach. So after 4 months, she was finally discharged, and she kind of pretty much recovered 90% of her baseline. And so at the age of 6, so we had about a good year and a half when she was, mostly seizure free. And then at the age of 6, she had another grand mal seizure. And this, again, put her under a induced coma. But this time, with all these different drugs that they're trying out, they tried out phenobarbital, and she looked okay for about 2 weeks, and we're about to get discharged. The day of discharge, she developed a fever, and also a high fever, and also a rash. And it took them 2 weeks to figure out what was wrong with it. And they determined, the whole team, after 2 weeks of trying to diagnose what it was, they determined it was a DRESS syndrome, which is pretty much an allergic reaction. It's a delayed allergic reaction, and that's how kind of made her look at that time. And this also caused the DRESS syndrome, and she also had some lung infections, caused her to have 3 cardiac arrests in the hospital. At which point they, administered CPR, and they were able to save her. And she has made a full recovery. However, her baseline has diminished substantially. So some of these photos, this was at the hospital, and then now this is more recent. Those 2 photos over there. where she's smiling and everything. So now at 7 years old, so our search, for a cure continues. Kendall's continued to get stronger. She's back at school with her nurse, para, ventilator, oxygen tank, feeding machine. And she's always happy to play with her two sisters. And I just want to thank you guys for allowing us to share Kendall's story. And hopefully it continues, and we'll give you guys updates. All right. That it?
If you could just wait there for a second. Okay, see if there's.
Hi, my name is Alexis. I'm a child neurologist from CHOP. Thank you so much for sharing the pictures and your story about Kendall. My question is, this morning we heard from a sibling of a child with a GRICon disorder, and I was just wondering, how did your oldest daughter kind of cope with all of these really traumatic and challenging events that happened in the course of Kendall's medical?
Yeah, so our older daughter is 9, so she's 2 years older than Kendall. And I think I don't know if she really fully understands, but we do have a child life specialist that would meet with our older daughter, Casey, about once every 2 weeks or every 3 weeks to kind of meet with her and talk with her and discuss certain things and give her certain situations and ask her, oh, how do you play with your sister and everything. And I think our older daughter is very, very sweet. I think she understands what Kendall has, not to a full extent, but she knows that she's not going to be, like her. Yeah. So.
Thank you.
Yeah. Okay. Thank you.
Okay, our next speaker is Colin Robertson. And I'm sorry, I don't have details on where you get to introduce yourself.
Okay, so I'm Colin Robertson. I'm a PhD student from the lab of Alex Velopoulos at the University of Maryland, Baltimore. And I work primarily on precision genome editing agents to create personalized patient models. There we go. Okay, so idea behind my project is to sort of expedite the process between bedside clinical care and benchtop research. We heard a little bit earlier about sort of the difficulties in creating patient models, the time and the financial costs associated with that. So the idea here is to use CRISPR genome editing as a possible intermediate solution. Idealized scenario is shown in the bottom left here. A patient walks into a clinic presenting with some sort of genetic disease, has their genome determined, pathogenic SNPs identified, and then CRISPR agents designed to directly model those SNPs. Then we can take those CRISPR agents, deliver them into wild type mice, and create on-demand patient models. Standard CRISPR techniques have a major drawback in this type of application in that they create double-stranded breaks, and the primary result of a double-stranded break is unintentional knockout of your gene. So in order to do this approach, I use a modified form of CRISPR genome editing called prime editing. You can see an example of prime editor here on the top right. It has two main differences compared to classical CRISPR-Cas9 approach. First is that the Cas9 is a Cas9 nickase, so it only cleaves one strand of DNA. It doesn't cause this double-strand break, so it drastically reduces those non-specific insertion and deletion events. And the second major difference is the fusion of this reverse transcriptase to the Cas9. That reverse transcriptase uses a modified guide RNA that you see here in orange to directly write desired changes into the genome. And this was first described just up the road in the Liu lab, and it was originally termed search and replace genome editing, and that's essentially how it works. So in our hands, we wanted to test the precision of this. This is a knock-in of a point mutation in hex cells. This is genomically in hexal genome. And the main thing to pay attention to here is the ratio of precise knock-in shown in green versus imprecise, unintended knockout of the gene shown in gray. These two samples here show the result of wild type Cas9. So this is traditional double strand break attempt to knock in a gene or variant of interest. And as you can see, the ratio of intended knock-in in green to unintended knockout in gray is completely unacceptable. We cannot use this in vivo. Compare that to the prime editing on the right, and now we've flipped that ratio of knock-in to knockout. So this is a much more precise system, and it gives us hope that we can take this, use it in vivo. All right, so my approach, again, is to use in vivo genome editing at wild type animals. This has a number of benefits. First is that I don't use transgenic lines, so it's all wild type animals. Drastically cuts down on the cost and the time to maintain and produce transgenic lines. Second is that I have flexible gene targeting. You can create an entire library of these genome editing agents and deliver them at will. Third is that the variant expression is endogenous, so we're not dealing with overexpression plasmids. It's in the tissue of interest. It's regulated by the standard gene regulators. And then fourth is that these are all independent editing events. So each animal model is an N of 1 animal model. So any non-specific off-target events that are happening as a result of gene editing are actually tend to be lost at a population level. And the goal for this is to go from detection of variant to a working animal model or some amount of an animal model within 7 weeks. To deliver these agents, I use a technique called in utero electroporation. This takes pregnant mice, you expose the uterus, and in the developing embryos, you can inject these gene editing plasmids into the ventricle, use electricity to force those gene editing plasmids into neural progenitor cells, and then those neural progenitors give rise to these excitatory neurons in the cortex that are callosal projection neurons. My specific variant of interest for these initial proof of concept experiments is GLUN2A A243V. This is an epilepsy-associated gain-of-function variant in GLUN2A. And I used a lab-developed web tool called Pegasus to design all of my prime editing parts. And you can see those parts here in the alignment to the gene. And then after delivery via IUE, I follow gene editing events at 3 levels of validation. So first in the genome using next generation sequencing, then in the tissue using whole cell recording and slice, and then third at the animal model using continuous EEG and EMG recording. So I don't have time to go over all of the data, obviously. Main takeaway is this seems to be a viable option. In a subset of the mice that were transfected and edited in this manner, we do see spontaneous seizures as a result of the patient knock-in. You can see here the waveforms corresponding to the seizure. The mice they're hard to see. This is not great video quality, but you can see the 2 mice there. This is actually the same mouse, 2 different occasions in the animal, and we will track across here. You can see the onset of a motor seizure, and this was observed in a subset of our prime edited mice, and it was not observed in any of our electroporated control animals. Yeah, and that's going to do it for me. Thank you.
Question right here.
For convenient. I have a question based on like the reproducibility of the mice.
Mm-hmm.
So we had an issue in our lab when we created a CRISPR model that because of premature death, we couldn't reproduce them and we had to end up using an inducible model. Is that something that you have like thought about? Because if you have a specific variant that results in premature death, you'd have to constantly go through this CRISPR process. So is that something that you've thought about?
Sure. So I think that's actually one of the benefits of my approach for delivery. So IUE, when you do this process, it hits about 1% of the cells in that layer of the cortex. So there's a really small population of cells. We actually didn't expect to see spontaneous seizures. We expected to see like small potential interictal events or seizure sensitization, but that was a surprise to us. I think the the limited number of cells that we're hitting is actually going to be kind of beneficial in preventing things like premature death in the animals. Mhm. Thank you.
Okay, next up is Brianna Kupras. And it looks like we may have time for a few extra questions at the end.
Hi, everyone. So my daughter has a Grin2E disorder, and different from most of the other families that we've talked to, she actually, her behavior patterns and her aggression is our biggest concern because it's the most isolating for us and the most, it affects our entire day and everything in our life. So, it's the big center one, right? This one, right? Okay, so Cece started displaying her self-injurious behavior around a year old. We started noticing delays with her at around 4 months. And then she started actually regressing at 6 months. So she stopped smiling, she stopped babbling. She was basically silent and non-moving for about 2.5 years. What she did do was hit her head on everything. So she still doesn't have a lot of function with her arms. She doesn't grasp things or eat independently with utensils. But instead of head slapping and face slapping that we see a lot of now after many years of physical and occupational therapy, she started by hitting her head and taking it to the ground first backwards, and then forwards on her crib when she was finally able to have enough core strength to sit up. We saw her taking her face to objects, which then changed us, changing her crib. We ended up padding a crib and basically making our own type of bed for her, because even the safety beds that are out did not really control the issues that we were having for her. Along with the SIB, she has a lot of aggressive behaviors. So hitting, hair pulling, destruction of property as of she's 4, so she throws things. It sounds worse than it is. But like shoving, headbutting, scratching, and biting. So all of these are things that happen all day, every day for her. So taking her in public has, the pandemic didn't help. So we were home a lot anyways, but now that the world has kind of opened back up, we still find ourselves very isolated because we can't keep her safe and we can't keep her peers safe as well. I swear I'm pushing the button. Okay, so different reduction in safety measures that we've taken over the past 4 years. We did try a soft helmet for her as she was taking herself backwards for protection. And that kind of became like a challenge for her. The more she wore it, the harder she wanted to hit. So we had to take that away. We did pad her entire crib when she started rocking backwards and then progressed to hitting her face on the railing and things like that. So now she actually has a queen size bed that we have in the corner of her room, and we've had to pad the walls on both sides about 5 feet high. So she does stand independently now and was going over top of the padding that we had. So now we have 2 layers of padding, and there is a picture. I thought my poster would be in here, but there is a picture of it out in the lobby if you're interested. So what we have taken is she has a lot of enclosed play areas with soft flooring. What we've had to do is carpet and pad the entire upstairs of our house so that she has not only her room and her space with all of her soft toys, but we tend to stick with like softer books and non-hard plastic things. So various medications that we have tried with CC to try to reduce some of the self-injurious behavior. We started first with 10X when she was about 18 months old. That was when she was diagnosed with autism. And her developmental pediatrician was actually the one who pushed for us to get genetic testing, because although she was experiencing a lot of ASD symptoms, it wasn't giving us any answers as to why she was regressing the way that she was. So we did see a reduction in the SIB, and that's actually when we saw a big increase in her aggressive behaviors. We did try clonidine, which they were hoping would reduce some of her aggression, but also help with sleep issues. The sleep issues were not related to the SIB. She had severe obstructive sleep apnea, which has been fixed with surgery. We did try NAC or N-acetylcysteine. We didn't observe any change in her behavior with that, although the NAC that we were trying, it came in a capsule that we had to bust open and mix with her food, and it smells horrible and it tastes terrible and it was disgusting. It was the worst 2 weeks of her poor little life. And right now she has been on Memantine, which she is a gain of function GRIN 2A variant. We've been using Memantine for 10 months now. We saw almost an immediate increase in her SIB. Sorry. And then about 2 weeks after, we saw a significant decrease in her SIB. So her highest recorded day of self-injurious harm topped out with 2,500 incidents in about a 14-hour period. So we thankfully have not had more than triple digits for SIB in over 4 months, which has been great. And it actually, a benefit that we weren't expecting was a huge increase in her communication. I do know that our neurologist that we worked with basically said the studies that they've had with autistic children using Memantine, they weren't looking specifically at one area. So it was kind of like, well, we don't see any negative side effects. You guys can try it and see if it helps. So her increased communication has been huge. She went from only having 3 word sounds to now she has over 200 words in her vocabulary. And within the past 4 months has started putting 2 and 3 word sentences together. Her favorite right now is telling me no thank you singing when I am singing. Yeah, it's fun. Yeah. So I do know that there are a handful of other GRIN 2A families that have also tried Memantine. So if you happen to see any of them around, you can ask them about their journey with that as well. And I do know that at my suggestion, some of our GRIN 1 families have also tried it, and they have had the exact opposite reaction where their child did not engage in self-injurious harm and immediately upon taking it started harming themselves. So it doesn't seem like what's working for us will work for other variants necessarily. So that is it.
One thing I'll just mention is that, oh, sorry, do you have more?
Oh, apparently I do. It's just isolating. That's our biggest issue. So I know that a lot of the families, you're still able to, with at least some adaptive equipment and things of that nature, are still able to maintain a fairly outgoing life. And for us, with Cece's particular variant, we have been it's very isolating. I like to call myself a lazy friend because now we have everyone come to us instead of us going to them because it's just safer for everyone that way. Yeah. So, and she's the youngest of 4. So we went from a very outgoing traveling family of 5 total to having her as our 6th in the family total. And we just took her on her first vacation ever a few months ago. So yeah, that's it. I swear now.
Thanks. If you just wait there for 1 second. I was only going to say that GRIN Therapeutics just dosed the first GRIN 2B gain of function patient, and they're planning to expand to GRIN 2A and GRIN 2B over the next few months. So not necessarily endorsing it, but we're talking to them to see if it's right for your family. Any questions?
Hi, thank you for your talk. I have a question about, have they done any, I would assume they've done EEG on your child?
Yes.
And a sleep EEG, just because we had a GRIN 2A family who had regression, and they found the spike wave.
That's what we originally thought that we were going to see when we had her sleep study. So the obstructive sleep apnea was actually a huge surprise for everyone. And it was so extensive that she had 3 millimeters of space for her airway after she was completely put under. So what looked normal upon just regular inspection, once she was fully sedated, everything just kind of closed up. But so she does have absent seizures, and she just started having nocturnal seizures, but we haven't seen a continuous spike and wave as of now.
Thank you. We have a new clicker. So our final speaker is, I want to make sure I say it right, Ludovica Montanucci. Is that right? Okay. With
Perfect.
Cleveland Clinic.
Yes. Can it?
Oh, sorry, that's the one.
Okay. Okay, I'm Ludovica Montanucci from the Cleveland Clinic, and I'm a researcher. And today I will presenting you a work about the prediction of and the interpretation of genetic variants. We all know that when we have a genetic sequence, it can impact at two levels. At the level of molecular level, where the variants can make a change in the protein structure, and this can affect the functioning of the protein. And also it's what we call usually functional consequences of the variant. And of course, at the phenotype level, phenotype can be health or disease. And as we know, many neurological diseases are associated to green variants. The problem is that the experimental determination of the functional consequences of variants is very expensive and time consuming. And most of variants are also associated with, classified as variants of uncertain significance. So it's important to have a computational predictor that is able to predict both the functional consequence and the phenotype. Also because, as you know, treatments like memantine functions only in case of gain of functions. So it's really crucial to know that. So our idea was to use the variants that we already know in order to build two predictors. The first one is a binary classifier, which classify variants of the green genes are benign or pathogenic. And the second predictor, also based on machine learning, which classify variants are gain or loss of function. So as you know, this is the representation of an NMDA receptor, which is the protein complex encoded by the green genes. And our first observation is, where do variants for which we know either the functional consequences or the phenotype are located in the protein? As you can see, you're probably familiar with the structure of NMDA receptor. It's made up of three different domains. As you can see, most of population variants, which are the variants that are found in general population, healthy population, tend to concentrate on the N-terminal domain, which is the upper part of the protein, while patient variants are mostly located in the ligand binding domain and transmembrane domain. The same, we can see a difference between gain and loss of function, while with gain of function variants located more on the helical bundle and transmembrane domain, while loss of function variants tend to be located more at the ligand binding domain. So we use this observation as a key feature in order to build our classifier. So this is the general design of our work. We used the larger data set of available association between variants and phenotypes, which is available in the GRICon portal, which you may be aware of. And we derived the population variants from the NOMAD database. We encoded this variant through different features. We use features for more that comes from the evolutionary history of the genes. We introduce biophysical features, so change in hydrophobicity and interaction energy between the wild type and the mutated protein, as well as residue solvent accessibility. And finally, we use the distance I was mentioning before, so the distance of the variant from all the ligands in the proteins. We merged and fed all these into a machine learning system, which will and we build a classifier after the training process, which is very laborious. So at the end, we came up with a classifier which displays 93% of accuracy. And this plot is a plot that shows the goodness of a classifier. All the, is there something? Yes, these lines are basically our classifier is the red line, the last line, and it is, let's say, the best one. A random classifier, this line indicates a random classifier, and the best classifier would be those that have the step function, a vertical line here and a horizontal line here. So again, this is for the pathogenicity predictor. We applied the same structure and the same design for the gain and loss of function predictor. The data set was smallest, as you know, because experimental determination of gain and loss of function is very laborious, but we end up with 92 loss of function missense variants and 84 gain of function missense variants in the green portal. We used the same input feature, and we came up with a loss of function predictor that has an accuracy of 80%. As you know, the lines are not so nice as before because we don't have enough training data, because this is much smaller than the one we have for pathogenicity. So in conclusion, we have identified that distance from ligands is a main feature in order to predict both pathogenicity and functional effect of variants. We developed the most accurate pathogenicity predictor for specifically trained for green variants, which enables also the reclassification of all the variants of uncertain significance. And also we developed the first functional effect predictor, also specifically trained for green variants. Wow. And thank you. These are all the people involved in the work, and thank you to everyone. And I'm happy to take questions and come to the poster.
We have a question.
Just a quick question. The data that you used the variants that you used to train the model, was it just grid in general, or did you try also like specifically like the grin 1, grin 2D, etc., or was it all just lumped as one?
So we merged them together. We did some methods in which we separate them. We were introducing some feature that were saying which variant belong to which gene, but basically it was not changing, because you see the structure. If we go to the structure of, can we go back? No, I don't know. Yeah. So if you see the structure, so green, green 1, you know, is this this color, and the blue, and the other one is the yellow. As you can see, they're very similar. So basically, it didn't make any difference nor improvement on the machine learning. We tried, but was not the case.
Sorry, it's just a quick comment. There is a publication that came out just January 2023 about this exactly, done by the group in Barcelona. So with 4,000 variants, including pathogenic and non-pathogenic, of course.
Yeah, they were using homology, I think, right? Or exactly distance in angstrom from ligands.
Yeah, I can send you the paper.
Thank you.
Yes.
Yes.
Okay, we actually do have a couple more minutes if there's questions. More questions for Dr. Ludovica or Dr.
For everyone else.
or for anyone, yeah. Oh, who's it for?
And it is Colin. So I had just a general question about the prime editing. Does it only affect one? It only fixes one strand of the DNA. Is that right? So does it matter? It does matter then presumably which strand you target it against, or does it not?
It's actually target either strand of DNA because the first transcript is just the right upper variant there, and then DNA mismatch repair will actually fix the other strand. So both strands will eventually.
So 50% of the time it takes it back, and yeah.
You can selectively pick which strands you want by the guide.
Right. Yeah. So I was kind of then wondering. I mean, because presumably at some point it either has to get it right when it copies it, or it gets it wrong. So I suppose if it gets it well, no, depends what right and wrong you mean. But eventually, if it makes it back to how it was in the first place, then it will just bind again, and the cycle will just keep on going until it actually makes the change on both strands. Is that?
Just realized people virtually probably couldn't hear your answer.
Yeah, so.
Yeah, so it's basically a 50-50 switch. You can target either strand of DNA with prime editing, and DNA mismatch repair will either incorporate your desired edit or remove your desired edit. And that 50-50 switch can be biased through the use of a secondary nicking guide RNA. So just an additional strategy to incorporate the desired edit.